which one's the, okay, thank you. Okay, so when I first came, I'm a memory disorders neurologist, and when I first came into the clinic, it was not long after we stopped defining patients by these parameters. Up until shockingly late in the 20th century, we would describe patients as having either senile or pre-senile dementia. And that really just meant that the patients developed their symptoms after age 65 or prior to age 65. And you can imagine why this type of classification was not particularly useful in defining prognosis or treatment trial groups. We got a little bit better over the past 2 or 2.5 decades, where we developed pretty specific clinical phenotypes that would define, we thought, certain pathologic phenotypes. And so we have very defined categories for what allows us to call a single patient an Alzheimer's disease patient or a frontotemporal dementia disease patient, to the best of our abilities, defining these patients as possible or probably having those diseases. And so during this time when we began to classify patients like this, we started a number of very high-profile, very expensive clinical trials, lots of them based on animal studies, and most of which failed. And that's mostly because, and this might be a slight oversimplification, that this is what these diseases actually look like. And so I should say that there are dozens or even scores of neurodegenerative dementias, so only putting a few up here is actually also an oversimplification. But the reality is that both the clinical phenotype and the pathologic phenotypes of these diseases overlap so much that by defining patients into these very narrow bins, we've likely done ourselves a disservice. So I'm going to talk a little bit about frontotemporal dementia today, and this is just one way to show you the complexity of this disease. So when a patient comes into my clinic and they're displaying certain phenotypes, and I'll talk a little bit about what those are in a moment, I might give that patient a presumptive diagnosis of FTD. They all look alike, but the reality is that those patients could have one of 4 different underlying general pathologies in terms of what their actual proteinopathy is, the most common of which are either a tauopathy or a TDP-43opathy. But then within each of those pathologic categories, there are sub-pathologic categories as well. And we used to believe that those categories then define the disease, but we know that that's not true anymore either, because a patient with Pick's disease, which is a behavioral variant form of FTD, might actually have, and very often does actually have TDP-43 and not tau, dysfunctional in their brain. So this is also likely incorrect. So I've told you that we originally categorized these patients in this fashion. We've gotten a bit better. That initially defined our attempts at rehabilitation, and we managed mostly symptoms. But we were able to add, through the efforts of a lot of years of very intense science, better structural imaging using MRI that has become more sophisticated with time, and now functional imaging. And this really encompasses both functional MRI and connectome studies. And that has allowed us to categorize patients a little bit better, but it falls down often still. So we've more recently added biomarker analysis to better classify these patients, including spinal fluid, and I actually use these in my clinical practice all the time. So I will measure amyloid, A-beta-42, tau, total tau, and phosphorylated tau in my patients, particularly when I'm trying to rule in or out an Alzheimer's disease pathology. Our imaging biomarkers have also come a long way. We have FDA approval for the use of amyloid PET tracers. The tau tracers are further behind. They haven't made it to market. There's too many off-target effects. But eventually, the hope will be that we'll be able to take a particular patient based on their clinical phenotype, check a number of these tracers, and look to see what actually their underlying pathology is without having to do a brain biopsy. Serum biomarkers are further behind. And then, of course, we can classify these patients based on their tissue diagnosis, a little bit like what I showed you on the FTD slide, and potentially by their underlying genetic disorder. But even that doesn't allow us to fully define the patient. And I'm going to give you the example in a moment of one particular disease in which that is not sufficient. So the reality is, it might be how that particular pathology affects that particular patient that's going to inform how we go after the disease. And that's really where the precision comes into play. And of course, there's a lens of an overlay here of all the other things that can affect that underlying pathology, including the patient's immune status, their genetic background, their microbiome, add your favorite modifier here. Okay, so I'm going to talk just briefly then about FTD and its sister disease ALS, or amyotrophic lateral sclerosis. So you guys may all know that disease as Lou Gehrig's disease. He, of course, was diagnosed at age 36 and died at age 38. And of course, that's a fatal progressive neurodegenerative disease. Patients usually pass away within 2 to 5 years. But the sister disease is this FTD, or frontotemporal dementia, which tends to come in 2 flavors, either a behavioral form or a language form, also universally fatal, and presents relatively rapidly and typically in patients younger than age 65. So what's fascinating about these diseases, despite the fact that they are clinically very distinct, is that they are genetically very overlapped. And so there are a number of genes that will cause both ALS and FTD, sometimes in the same patient. And that really is a spectrum. So this is incomplete, the actual genes that cause these 2 diseases. There are upwards of 20 or 25 of them, and somewhere around 8 or 9 that will actually cause an overlap syndrome relatively frequently. So what I alluded to before about how the genetics may not necessarily inform the clinical presentation is really defined here. So in C9orf72 is the most common genetic form of both ALS and FTD. And in the same exact family, patients can either get ALS, FTD, or both. And we really have no understanding of what the factors are that lead to a patient developing one or the other. And we certainly don't know if the therapies that we currently have or are developing will attack whatever part of that pathway leads to both of these diseases. And certainly the cortical and the motor neuron phenotypes are very different. So when we're studying these diseases, of course, with the increasing simplicity of our models, we have decreasing relevance. And so a lot of the focus in studying both ALS and FTD has been in the mouse model. And I would argue that potentially for the spinal pathology, that's really relevant and can lead to a lot of discovery. But in an animal model that doesn't contain particularly developed frontal lobes, it may not be as relevant for the cortical neuronal phenotype. And so thankfully, we have an intermediate stage now, since we can't go study directly in our human patients. And so our lab is very interested in using stem cell models to study these diseases. You're all familiar with the fact that stem cells can be created from adult patients using either peripheral mononuclear cells or fibroblasts. And those induced pluripotent stem cells can be used to develop and derive any lineage, really, depending on the complexity of the protocol. And I'll draw your attention just briefly to this lifesaver-looking cell here, which is your neural precursor cell, which will go on, if prompted correctly in the lab, to develop both neurons and both types of glia, and of course not the other resident cells of the CNS like microglia. But effectively, you can take these cells and grow a brain in a dish. So the eventual hope is we'll take cells from patients like Sabrina. So Sabrina was 16 when she passed away from familial ALS. Derive stem cells from patients just like her, and then drive those stem cells towards the neuronal fate, create these brains in a dish, and use those to study the disease process, which is what my lab does, but also potentially to screen specific drugs for that particular patient. Of course, the other ultimate hope is that you might be able to correct the genetic deficit in that patient's cell, then grow essentially a neuronal graft and transplant back into the patient. Now, this has actually been done in mice that contain the SOD1 mutation in ALS, where they were successfully able to use the CRISPR technique to fix the genetic defect and then transplant motor neurons back into the spinal cord of that mouse. For neurodegenerative diseases potentially with a prion-like transmission mechanism, that may not be as useful because the transplanted graft may just become diseased. But that's a discussion for another day. Okay, so most of the people who study stem cells then study them in our typical cell culture fashion in their lab. So In other words, studying those neurons in 2 dimensions. And our lab doesn't. Our lab studies these cells in 3D, and there have been some great papers that have set precedents for why this may be superior and more relevant to the physiologic state. I've listed a few of those here, but I just want to draw your attention to the one that we think might be most important, which is that many of these neurodegenerative diseases are diseases of secreted proteins. And when you grow these cultures in a hard dish and change the media every few days, you're of course washing away all of those secreted proteins. And in addition, when you're talking about how cells respond to a drug, when you're bathing 50% of the cell surface in the drug in the media, we know that the cells don't respond the same way as they would in a complex environment with dense extracellular matrix and cell processes. And that's been very well established in the cancer field. So our lab studies these cells in 3D, as I mentioned. So we take patient cells, we derive our neural stem cells, and then we seed them in one of 2 3-dimensional formats. From there, we studied these 3D neural cultures for viability, morphology, cell deficits that might be targetable in terms of drug screening, and then developing those platforms for future drug screening. So I'm out of time, but I just want to summarize by saying that the ultimate goal here in terms of precision neuroscience for neurodegenerative disease is to identify these patients before disease onset. We can do that now to some extent using some of these biomarker tracers to accurately diagnose the patients using the techniques that we've talked about, and then potentially tailor therapeutics specifically for those patients using either their own cells or understanding the biology in a way that informs the treatment. That's it.